I'm Rebecca Sweat. I'm one of the co-chair organizers for BAGIM over here in Boston, and welcome to our BAGIM-SAGIM joint seminar hosted by both the Boston Area Group for Informatics and Modeling and the South California Area Group for Informatics and Modeling. And yeah, so first up, as always, a giant thank you to all of our sponsors. Thank you so much for this. Schrodinger, OpenEye, CompCam, Crescent, Informatics. I won't read them all off, but if you'd like to participate, your name or logo could be right down here. If you feel like donating or helping us continue on our wonderful seminar series, it's all volunteer run. If you are interested in becoming a BAGIM sponsor, please feel free to contact Tony at arvanites@bagim.org. So we have different levels of sponsorship. So at the silver level, you have recognition at meetings and online. At the gold level, you can share a session, and at the platinum level, you can have your own sponsored non-commercial speaker. So you get to pick the talk, which is great because that means that we don't have to organize it. We, of course, would host it. All right, so just so you know who we are, this is the Boston Area Executive Committee that consists of Brian Kelly, Tony Arvanites, Scott Parker, and myself. And joining us tonight are our SAGIM Executive Committee, Jamie Schiffer, Scott Lyon, Tony Ivitak, and David Slotschauer. So with that, I'm going to stop talking to you all so we can get to the really interesting part of the night. And the first up is Doree Sikoff. She is the principal scientist in CompChem Discovery Chemistry at Janssen, and she is going to give us a wonderful talk. So I will stop sharing so she can share. All right, so Doree is unmuted and ready to go.
Okay, so we're having an issue here. One sec, let me do this again. Okay, try again.
Let's see, can I ask to?
All right, is it working
There you
now?
go.
Yay!
Looks great, looks great.
Okay, sorry about that.
All good.
All right, so hi everybody. I'm really, happy and thankful for this opportunity to talk to these groups. So my name is Doree Sikoff. I am in the computational chemistry group at Janssen. I've been there for about 1.5 years, and prior to that, I was at Bristol Myers Squibb for a little over 20 years. So today, what I'm going to talk to you about is applying torsional health assessments based on crystal structures to ligand docking. And I need to advance my pages. Not working, there we go, okay. Okay, so I'm actually gonna advance a couple of pages. Okay, so, I come from drug discovery in the pharmaceutical industry, and so this is my introductory slide. This is what my introductory slide looks like. So this is showing a big range of computational chemistry technologies that we employ across drug discovery to bring a potential drug molecule from target ID all the way to late lead optimization, from which it would then go into clinical trials. So these are a lot of methodologies that all of the modelers are familiar with. And what I wanted to point out here is that in a great many of them, the ligand conformations play an important role in the technologies. And so it's pretty important that our ligand conformations that we're using be accurate. So what are the sources of the ligand conformations that we use in modeling? Well, they come pretty much from 3 sources. They come from an experimental or knowledge-based source. So that would be things like small molecule crystal structures or co-crystal structures in the PDB. They come from force fields, which is based on classical modeling, and they come from QM. And often they're coming from kind of a mishmash of these things that are all being used together. And so what I'm going to go through in this talk is that my argument would be that special value should be given to the experimental or knowledge-based data because it's real. And, we could quibble about that. We could say that if these are coming from a small molecule crystal database, is that really the same thing as what we would expect to be going on in a PDB, in a protein ligand co-crystal structure? I don't know, but we could talk about that later. But for now, I think it's certainly valid to give this experimental version some weight. Okay, so what I'm going to talk about in this talk is a way that we've gone about flagging unusual conformations. So that's relative to the experimental data to inform our modeling results. Okay, and so now I'm going to go back up to, I apologize for this. I lost my final version. So, okay, so now I want to go through the outline. So I just gave you the introduction. And what I'm going to talk about next is this method that we're using, which is to utilize Mogul, which comes from the CCDC tools, to calculate a geometrical health grade for ligand conformations. And then I'll talk about trying to apply that to two applications in ligand docking. So the first one will be pose assessments. So trying to use this tool to help us rank which poses might be closer to the real way that a molecule binds into a protein. And then the second application will be another way that we use ligand docking, which is in virtual screening. So to try and enrich the number of hits that we'll find in a set of molecules that we test in an assay. And usually we identify that set by ligand docking, and I want to see what happens if we put this health grade filter on top of that. Okay. Sorry about this. Okay, so here I'm going to talk about CCDC's Mogul tool, and I'm hoping that a lot of you are familiar with this tool. So Mogul is a knowledge-based library of molecular geometries that are derived from the CSD, the Cambridge Structural Database, which is a database of small molecule crystal structures. And so what Mogul can do is identify unusual geometries in a given geometry, in a given conformation that you feed it. And the way that it works is, so for this, my query 3D molecule, so say I'm focusing on a torsion that involves these 4 atoms, it compares the torsion that's found in my query molecule to the torsions that are observed among the molecules that are in this crystal structural database. And so that's what's shown in this graph, all the torsion angles that could exist are down across the x-axis, and then the y-axis showing how many times those particular values were found among the similar sets of 4 atoms, or within those groups that are in the database. And so the way that Mogul picks which are those similar groups in the crystallized small molecules is by using some generalization rules to identify which ones are similar. And then, so what's shown in this graph is an example. So you can see these 2 peaks where there's a lot of observations of those torsion angles, and this query, you can see by the red line, lies well outside of what's found in the database. And so in addition to giving out graphs like this, Mogul can also output textually. It can tell you that this query, the value in this query is unusual based on what it's found in the database. And the definition that it uses by default is that if less than 5% of the examples in the CSD are within plus or minus of the query value, then it's identified as unusual. So again, so if there's less than 5% of the examples in the CSD within plus or minus 10%, then it's unusual. So that kind of makes sense, right? I'm going to get my pointer. Okay. Okay, so, all right, so then I'm up over here. So there's, some kind of issues. So if we consider this kind of flagging unusual torsions idea, there's, some issues that you might see about that. It's just it's a yes/no rule. And so there's no sliding scale for whether this line in the query was like really far away from all the observations or just far away, right? So there's no like graduation in that. And also, you're limited to what's available in the library. So if you have a query molecule that has groups that are just not observed, not what's been crystallized in the small molecule crystal database, so be it. Okay, so going to the next slide. Now what do I do? Okay. All right, so so here's just an example of this unusual torsion count, which I want to propose for assessing the geometric health of a molecule. So we have this molecule, this ligand that's shown here, and when we run it through Mogul, it identifies that there are 2 torsions in this molecule that are unusual. So by that filter of 5% outside of plus or minus 10, those meet that criteria. So we could say that the score for this molecule is 2 total unusual torsions. Okay, so here's the tool that I'm proposing that we've put together, we've scripted it. So what happens is you feed it the 3D structures, and then the tool runs them through Mogul. And then from the output from Mogul, there's kind of some massaging that goes on, but you end up with a count of unusual torsions. And then that value goes into the properties, it gets put into the properties of the molecule. So what comes out is a structure file that has the properties, these new properties in it, which can then be analyzed either by like filtering the flat file or by reading the file into some kind of GUI where you can look at the structure. And so the properties that we put into the file are the total unusual torsions and also the atom IDs that are involved in the unusual torsions. So then, if it's, a GUI that you can work with, you can, write some macros or something to get it to render those bonds differently so that you can see which are the torsions that are at issue here. And so I also wanted to note that this tool that we've got here, it's like a bunch of scripts, and the timing of how quickly it runs, it's not blazing fast. It's 4 structures per second, and that's on a Linux box Linux workstation that I have at my desk. Okay. So all right, so we have this tool, and we want to kind of see, is it actually, is it useful? How does it do if we try and run it against crystallographic ligands? So we know that it ought to work pretty well if we used small molecule crystals, right? Because it was derived from the small molecule crystals. But how will it do if we try and feed it crystallographic ligands from PDB complexes? So I would a priori expect that crystallographic ligands, even from the PDB, should have a few unusual torsions. And again, that might be something that we could argue about because, are small molecules really relevant to PDB structures? Small molecule crystal structures typically have high resolution. There's more packing perhaps. But I'm using for this test case, I'm going to be picking out some very special PDB structures. So this is the Iridium highly trustworthy data set that was put together by OpenEye a number of years ago. There's a reference. And they picked out 121 co-crystal structures with really strict criteria, which are listed here. So these are meant to be very high quality crystal structures where all the density is present for the ligand. And so that's the data set that I'm going to use. So what I do is I pull out those crystallographic ligands and I feed them through the tool. And this is the result that I get. So I have the number of unusual torsions across the x-axis and how many ligands were observed to have that many unusual torsions. And what you can see is, it certainly does cluster towards the lower end of unusual torsions, but there's actually kind of more unusual torsions than I think there ought to be for crystal structures. And so one thing that we were thinking is that maybe it's this criteria of 5 within 10 that might work fine for small molecule crystal structures with high resolution, but what if we relaxed that criteria a little bit to 5 within 20? So this is 5% have to be within plus or minus 20 degrees. So that's a much wider window. And so when we do that, though, those are the orange bars, and you can see that you get a whole lot more that are at 0 unusual torsions, which is kind of comforting, and fewer that have higher unusual torsions. And so you end up actually, if you use this definition, you get 97% of that highly trustworthy set with unusual torsions less than or equal to 2. So that's a kind of cutoff that I'm going to go back to a few times during the talk. And then I just wanted to show you a couple of examples of these torsions that flip, whether you use the 5 within 10 or 5 within 20 criteria. So here's this example where I've labeled the unusual, I've rendered the unusual torsions if you use 5 within 10 versus 5 within 20. And so 4 of these go away. And these are the 4 that go away. And you can kind of examine them and see if you think it's fair that they go away. Like this one, for example, it isn't a minimum, but there's still quite a number of structures that are present that have that torsional value. And then in the next slide, I have the one that remains. So this is the one that still is identified as unusual, and I think everybody would agree that's probably unusual. Okay, so now I want to get into the applications of to ligand docking. So this is again my first application of wondering whether we can use this this kind of grade, conformational health grade, to help us with identifying which docked poses might more closely resemble a crystal structure if we could get it. Okay, so this, so before we get to trying this out, just thinking about this, it's kind of obvious that it would be useful for a modeler when they're looking at poses to try to have a count or to have some assessment of how many unusual torsions there are in that conformation. Right now, a lot of the modeling software has a link into the Mogul tool, but what it shows is kind of the whole picture of the molecule, and it's difficult to parse that out. The modeler kind of has to look at it to figure out what's going on. Whereas if you just have this single count that you can put in a column next to the docking score, it enables you to see what's going on more quickly, maybe to reorder things based on how many unusual torsions there are. And that's what I'm kind of showing here, so that you might have docking scores that are really essentially equivalent to each other. And so you want to look further to try and determine which of these docking poses might be the correct one. Okay, and then there's one other point here, so besides helping you to figure out which pose might be best, even if you find a pose that you think might be the bound pose, if there's an unusual torsion, that's a spot that you want to be able to highlight, like this rendering does, because that's a place that you might be able to modify your ligand and get even more potency when you bind to the protein. Okay, so now I want to actually go to another test case, another experiment, and see if adding this filter on really does help you identify the right binding pose. And for that, I have to use a case where I know what the right binding pose is. So I'm going to try and dock ligands back into their native crystallographic proteins. And what I'm going to use in this case is the DUDE data set. So a lot of people are probably familiar with this. There's about 100 protein targets, and there's one protein crystal structure for each of the targets with an associated ligand, and it's ready for pretty much, kind of ready for docking. There's some other features of the DUDE data set that will become more useful when I get to my second docking experiment. Okay, so the protocol that I'm going to use is I'm prepping a protein. I'm using lig prep, or I'm kind of Schrodinger heavy for the ligands. Glide SP is the docking method that I'm using because I'm trying right now to identify a docked pose. And what I'm going to try are 5 poses per input structure. And since I've done lig prep, sometimes there's more than one input structure for a particular ligand because it might be a tautomer or a protonated form. And I want to give a shout out to Nathan Lim, who's a postdoc in our group, who was the one that pulled down these structures originally and did a lot of calculations. And I've also just got here in the corner showing what the unusual torsions are for the actual crystallographic conformations. So this is kind of the best that we could probably do with our docking poses as far as getting something exactly like the crystal structure. If we got a pose that was exactly like the crystal structure, this is what we would get. Okay, so here's the results that I get. Again, this is for 5 poses per input structure, and I've actually put in a cutoff for the docking score, since we're trying to get poses that might be the crystallographic pose. So those we would expect.
I got a question.
Yeah. Question, yeah. Or maybe we're leaving questions to the end. I guess maybe we're leaving.
Yeah, let's leave questions to the end, and please put them in the chat.
Yeah, I think that was Gregory.
Okay. Okay. So of these poses that we're trying to look through and figure out which one might be the right one, I have the results here. So I'm plotting the number of unusual torsions on the bottom from 0 to 5, and then the RMSD from the true crystallographic ligand up here from 0 to 16. Sorry, the numbers are kind of small. This line is at 2 angstroms away from the crystallographic structure. And what you can see is that as you get to
Janssen.
As you get to the larger numbers of unusual torsions, you find fewer poses that really match the crystal structure closely. So that's one pattern that you can see here. And then I'm also going to make the argument that these particular hits, so these poses that had 3 unusual torsions and still were close to the crystal structure, those are listed here for one of the targets. And I would argue that we could actually have ignored those as well, because in this case, there was another pose that actually I drew it wrong. It was actually in the 1 unusual torsion. So this was a pose that only had 1 unusual torsions instead of 3, and it still had about the same docking score. So if I, didn't know the right answer and I was just looking through these, I would probably throw,
Yo, can we pause? I gotta take a shit.
Okay. I would probably deprioritize these ones that have 3 and prioritize the one that is 1. Okay.
Hello, did you fucking hear me?
Can we mute that person?
Yes, I'm gonna remove them.
Can we also force muting?
They're gone.
They can't unmute.
No, they're completely removed from the meeting. That person's gone.
Thank you.
Yeah, I don't know who that was.
Okay. All right, so let me quickly get to the second application, which is whether we can add this filter on to try and enrich our virtual screening hits. All right, so again, I'm at the DUDE data set. And so
we are farmers.
So what's interesting about the set is that there are, for each target, there are known actives. And then the group that put this together has also identified decoys for each of the target that have similar physicochemical properties to the actives. So that's kind of our screening set that we can look through and see if we can increase the number of true hits that we identify. So for this case, I'm doing the same docking protocol, but I'm only using 1 pose per input structure, because that's the protocol that you would usually use for virtual screen of lots and lots of compounds. I'm going to put this filter on of total unusual torsions less than or equal to 2, and I'm going to look at the change in enrichment factor when I use that filter or not. And so here's the results. Definition of the enrichment factor is the fraction of true hits in the set that I picked to test, divided by the fraction of true hits that were in the full data set. So how much did I increase my chances of getting true hits? And so the results on the left are shown for what I'm plotting here is the enrichment factor from docking alone across the bottom. Oh, and this is only for the top 1% of hits that I'm picking out. Okay, so the enrichment factor from docking alone, and then on the y-axis is the change in enrichment factor that happened when I added on that filter. And what you can see is that most of the dots come out above 0, so we're getting enrichment from putting in that unusual torsion filter. It's small, but it's there. And then on the right, I'm plotting just the targets, like kind of just in a line, but separated out by the target class. Just to see, I guess the kinases seem to, I'm not sure that there's really a correlation there, but it's just kind of interesting. So that's the end of my talk. You know, I think we're in agreement that unusual torsion counts can be an aid to modelers for a number of reasons, for interactive use, and then also potentially in virtual screening. Although I think some more work has to be done there. So really, we'd like to apply it to some other docking methods like the high throughput virtual screening. And then I think that the tool could probably also use some refinement, for example, for molecules that have a lot of rotatable bonds. And so lastly, I'd like to acknowledge the Janssen computational chemistry group, some of whom are written here explicitly, especially Nathan, who did the dude calculations originally without the Mogul on top. I'd also like to acknowledge the group at Bristol Myers where I was previous to Janssen, where I first implemented a previous version of this tool, and also the CCDC support group. Thanks, everyone.
Great, thank you so much, Doree, and I'm so sorry for the interruptions. The meeting is now locked and those two are banned. So do we have any questions in the chat? Does anybody want to ask Doree anything at the moment? I mean, I myself have one pure curiosity question. What on earth was that molecule with the 8 unusual torsions?
Yeah, I actually think that there's something wrong. Well, as you might guess, I think that there's something really wrong with how the bonds were assigned or something in that molecule.
Gotcha. All right, we have a question from Lee in the chat. Is there an easy way to identify unusual torsions in docking poses?
Yeah, so that's what this tool is doing. If you run the pose through my tool, then you come out with a value, which is the number of unusual torsions, and the atoms that are involved in that unusual torsion are noted so that you can read it into whatever GUI you have and observe them and see them there, if I'm understanding the question correctly.
Yes, we have 2 more questions, and then we're going to move on to our next speaker. What do you think of including QM generated torsions to build up the data set above what's in the CSD?
Yeah, I think that would be a good idea, especially for torsions that are missing from the CSD. I think that would be great. Yeah.
And I'll take the last one from Callum. How does unusual torsion count correlate with glide internal ligand strength?
Yeah, I was asked that as well. I have to go back and look at that and see. That's a really good question. I've been asked before. I haven't had a chance to look at that.
All right, for the sake of time, we are going to move on to our next speaker. This is Greg Warren, the Director of Computational Chemistry at DeepCure. And I will have all of the questions in the chat. I'll record those. So if feel free to reach out. If you want to ask Doree more questions offline, we can maybe see if we can arrange that. All right. Greg, feel free to take it away, and I will ask you to unmute yourself.
Did I unmute? Yeah, perfect.
There you go.
All right.
Great.
All right. So thanks so much for Doree for introducing Iridium, because now we're actually going to talk a little bit more about it. In many ways, we probably should have reversed these talks. I also need to thank Doree for allowing me to steal her title. So I'm going to talk about sifting through the PDB instead of sifting through poses. I'd like to thank CCDC and the organizers for BAGIM and SAGIM for letting me talk this evening. I do know a number of people have heard this talk before. So for those of you who were thinking of taking a nap or maybe watching that YouTube series that you had all queued up, I'll give you a slight challenge. And it is as follows. For the sake of the narrative, I've included results here that have a slight scientific problem. And if you can identify that data set and what the problem actually is, you earn a free drink paid for by Scott Parker. Just a minute. I'm sorry, I forgot. I didn't approve that Scott Parker thing first. So before we start the talk, I'd like to go in and talk about a little bit about structural biology and comp chem, because Doree did talk a little about crystal structures. So I'd like to point out, it's important that we remember that protein crystal structures are models. The data that's collected from diffraction, whether or not it's X-ray diffraction or neutron diffraction, is essentially intensities, a diffraction pattern of intensities. And that diffraction pattern is then turned into a model of electron density. The electron density isn't data. It requires information for the most part for us to model it from the diffraction pattern. We then build coordinates into that electron density. Those are again not data, they're a model. And then when we run our calculations, we're running models on top of models. So it's important to remember that if we're not paying attention to the underlying data, then there's going to be problems with our models of models. So the outline today is talking about what problems there are on the protein ligand side, talk a little bit about iridium. I'm going to then talk about some results, what happens when we look at the data and how does that affect our results, and then there'll be short conclusions. In 2012, Ant Nichols, Brian Kelly, an undergraduate from Gonzaga University, my brother and I published a paper on how to go through and select protein crystal structures for use, in particular, evaluating them and figuring out which ones are good or bad. In the same year, the CCDC also published a paper on this topic, and a year later, Bernard Rupp started the series of papers which are titled Twilight, again on this same subject. This is carried on, and what I'm showing you here is actually a cut from a preprint that's being put together by David Hahn on how to choose selective structures for benchmarking free energy methods. And the criteria here are very, very similar to the criteria in iridium. It's just, I wanted to show you that these are the criteria that we used and thought up on how to select crystal structures. So prior to 2012, most of the people, when they went through and collected crystal structure data sets, used resolution as the criteria by which they selected those structures. And there's a couple of reasons for this. One is that they believed that resolution was actually a good metric for selecting crystal structures. And another one, which I think may have been unconscious, was that it's an easy metric to grab. It's right there in the title. You can grab it, you can filter it, it's easy to use. But there are problems with that. And what I'd like to show you here are two crystal structures. So one on the left, one on the right. And the question I would ask is, which one do you think is higher resolution? Is the left one the highest resolution, or is the right one the highest resolution? If most of you are thinking the right one, that's actually a trick question, because these are both identical resolution. And the problem with resolution, or what I'm trying to illustrate with this slide, is that resolution really doesn't tell us about the quality of the model, and it doesn't tell us about the completeness of the data set. And in fact, the structure on the left has an incorrect ligand conformation and really shouldn't be used if you're trying to model something off that ligand conformation. Well, you might be saying to me, well, the problem was that you chose a 2 angstrom structure. If I choose higher resolution structures, this problem will all go away. So let's look at a higher resolution structure. And here you can see that the ribose conformation is actually incorrect as indicated by the difference density in this structure. If we remodel this ribose conformation and remove that water, we get a new conformation that is actually correct. There's no difference density, and it's very, very different than the one that was deposited. So high resolution isn't always an answer to the problems. Very high resolution isn't always an answer to the problems either. Well, there are other metrics that we can use. Maybe we can use a metric that tells us whether or not the model's good, like R factor. For those of you who don't know what the R factor is, it's basically just the difference between the intensities, of diffraction intensities, and diffraction intensities are calculated from the model that you're refining against. And the smaller the difference, the better the structure, the better the model. The problem is that we actually can't compare R factors between different data sets. One way to think about this is that R factors are a lot like the integration of NMR peaks. They're relevant within the same data set, but they're not relevant across different data sets, or they're not comparable across different data sets. So if resolution and R factors aren't a great way to do this, what is a better way to look at ways of filtering the structures that we want to use? Well, one thing a sidelight I'd like to point out is that coordinates aren't infinitely precise. And I'm showing you here by looking at anisotropic ellipses that even within a crystal structure, the precision of different atoms varies. And one of the things that we have to worry about when we choose a global metric is that masks the variability within the structure. And if there's a high amount of variability or less precision in the active site, that could be a problem. But nonetheless, it is good to have global measures. The problem is that resolution doesn't translate into a understandable way of understanding the noise, the precision of the coordinates we're actually working with. In 2002, David Blow generated a mathematical formula to estimate what the coordinate error was from a crystal structure. If you look at this formula, you can see R factor, fit to the model is a component here. The number of heavy atoms in the crystal structure or in the asymmetric unit is a factor. The volume of the asymmetric unit cell is a factor. And the number of diffraction patterns recorded or number of observables is also a component in estimating the coordinate error. So now you have a measure of the model, the measure of the completeness of the data as a way of estimating how precise the coordinates are for this structure. If we look at a comparison between, on the x-axis, resolution versus coordinate error for a number of crystal structures, what I'd like to show you very quickly here is that for different bends, I'm showing you bends at 2 angstroms, 2.2 and 2.5 angstroms, there is a high amount of variability in the coordinate precision. In fact, about 0.5 angstroms variability between different structures in these same resolution bends. But even more disturbing is this example right here, where we have a structure at 1.8 angstroms resolution and a structure at 2.8 angstroms resolution which have exactly the same coordinate error. Now, if you grab that 1.1 angstrom structure, you'd think you'd be using a great structure. It turns out it's actually not a great structure, and that 2.8 angstrom structure is just as good as the 1.8 as far as this global metric is concerned. So just to summarize the global measures, resolution really is a bad metric of quality. You should use something like coordinate error. R factors, there's no really good single value, but there are poor values. So you need to watch out for poor values. And what I haven't mentioned in here is that one of the things that's important is the ability to assess a structure only occurs if you actually have the experimental data available. So if the density or if the diffraction pattern isn't available, it's pretty much impossible to assess the quality of the structure. Now let's look at some local measures, because when we go to do docking or we go to do simulations, we care about how ligands or the protein is interacting in the active site. That's what we're modeling. And some of the things that we need to pay attention on the local level is things like crystal context. These structures are packed into ordered arrays, and sometimes one protein interacts with another. In particular, if it interacts with the active site, that could be a problem. I'm showing you an example here of 2 phosphate atoms that are actually 4.8 angstroms apart, not something you would expect to see between 2 negative charges. How is that possible? Well, it turns out there's a crystal packing partner that inserts a nice little lysine between those to form a salt bridge, between those two phosphates. And were that lysine not there, the ligand conformation would probably be very, very different. Another thing that we have to worry about is whether or not the side chains that we're using to calculate interactions are actually modeled correctly. So we need to make sure that the active site density is complete and that the side chains that are being modeled are correct. Show you example here, the ligand is very well defined with electron density. The bottom part of this active site pocket is well defined, but we have a glutamic acid that's interacting with the ligand, and you can see there's a little bit of density for the carboxylate and nothing in between, even up including the backbone. So whether or not that glutamic acid is there, I don't know, but there's certainly not experimental data to verify or confirm that. Another thing that we need to worry about in structures is if there's alternate conformations. This isn't a bad thing. It actually says that the data quality is good and the modeling has been done very well. But the problem is how we use these. If I have an automatic method, preparation method, that takes only one alternate conformation and throws away the others without assessing them, that can be a problem. Let's take this example, for instance. I have two alternate conformations. If I grab these two conformations, I now have the ligand clashing with a side chain, and that's not going to work out very well if I'm trying to dock a ligand back into here. So one of the things we need to pay attention to is whether or not the alternate conformations are coordinated, and select also the alternate conformation that has the highest occupancy. That's the most prominent one, the most dominant one in terms of the conformations that we are going to be working with. So to summarize the local measures, we need to make sure that there are no crystal contacts near the active site. We need to make sure there's complete density for the ligand, complete density for the active site residues. We want to make sure that there are no active site residues with low occupancy. We need to make sure if there are alternate conformations that we've selected a conformation with the highest occupancy and the least clashes. We need to identify covalent ligands. This is particularly a big problem for covalent docking, but also in reality could be a problem for simulations as well if that covalent ligand has modified the conformation dramatically. And for simulation methods, we need to make sure that there's no missing side chains or loops near the active site, because if we model those back in, we are perturbing the experimental data in ways that we don't know or understand. So now that we've talked about how we go about selecting the data or filtering the data, what happens when we use this data? And I'm going to show you 3 examples of the results of using data. I'm going to talk about a post-prediction example, virtual screening example, and also talk about ligand conformations. So in the first case, what we're trying to do is assess small changes in a scoring function. So at OpenEye, there was a change made to the docking scoring function called Kim-Gauss3, in which in Kim-Gauss4, the hydrogen bond geometry calculation became more physically rigorous than it had been in Kim-Gauss3. Now, when we measure this change, we're going to expect a priori that Kim-Gauss4 is better. It's a better physical representation of chemistry and physics. But we would expect, since this was the only change in the scoring function, that the difference that it's going to make would be very small. So when we generate a data set to test this, we need to make sure that it's a data set with very high confidence, because otherwise, if there's a lot of noise in the data set, we're unlikely to see this small difference. So what we did is we took, again, the Iridium HT data set that Doree mentioned, and we did cognate docking for the ligands using Kim-Gauss3 and Kim-Gauss4. We measured the correctness of the top-ranked pose using a shape metric, so Tanimoto combo, and we also used the standard RMSD. You can see that if we measure the significance of the difference between those, we have statistically significant difference if we use a Wilcoxon p-value. We did not have it for the t-test in RMSD. It's important to note that the Wilcoxon is probably a better statistic here because the t-test assumes a normal distribution and RMSDs aren't normally distributed. So from this test, we can say definitively that CG4 is different than CG3, and we assume that it's better and that we're moving in the correct direction. But what would have happened if we hadn't been very careful about how we select the data? What if we went back and grabbed the Iridium moderately trustworthy data and ran the same calculation? we would have found no statistical significance between Kim-Gauss 3 and Kim-Gauss 4. So we would have never been able to prove that that change to the scoring function actually created an improvement. I will point out, though, while I've showed statistical significance, the difference between these two is extremely small. So there's no substantial statistical change in these two different methods. Again, we expected that. We expected the difference to be small. So we don't have a substantial change. And if we want to see substantial changes to the docking scoring function, we're going to have to add other things before we're going to get a substantial improvement. The second case is a virtual screening case, and we're looking at MAP-K14 or P38. This is taken from the DUD data set, not DUD-E, because this work was done in 2008. I was digging back through my archives of data and found this example. When DUD was released, the protein that they recommended that you use was 1Kv2. If we run an assessment on this crystal structure, it comes as a not trustworthy or not applicable crystal structure. The reason is that there's no deposit electron density, so we can't actually assess the quality of the structure. But if we estimate the coordinate precision, you can see that this structure has a coordinate precision of almost 1 angstrom. There's a lot of noise in this structure. So we went looking for some other crystal structures, and we found 2ZB0. It's an MT structure. As an alternative, it has a coordinate precision of 0.26 angstroms. We also found D83, which is an HT alternative. It has a coordinate precision of 0.24 angstroms. So what happens when we take these 3 different crystal structures and do the virtual screening test using the DUD data? Well, you can see on the left here for 2Kv2, the NT, I'm showing you the AUC values. In the blue bar, that's the docking score, you can see that the AUC is barely above random. And in fact, if you look at the error bars fall below 0.5. So this is probably a random result if we look at statistical significance. If we do the same calculation with the MT structure, you'll notice there's a statistically significant improvement where we move above 0.6 in the AUC. And if we go over to the HT structure, we see another improvement again, where we're up to about 0.66, 0.67 for AUC. So the difference between the NT structure and the AT structure is not only statistically significant improvement, but it's actually a substantial improvement as well. So just to summarize this about quality in versus quality out, to assess very small changes, we need high confidence data. Hopefully I've shown you that in the first example. If we use high confidence structures, those can improve virtual screening performance, at least in this one example. So the last example I'd like to talk about is ligand conformations. You've all seen local ligand strain papers and looked at, I'm showing you a histogram of ligand strain taken from a data set of 183 crystal structures, 186 crystal structures. And you can see if I calculate the local strain from the raw PDB coordinates, I have a mean value of 35 kilocalories here. Now, what we were interested in this study was not necessarily the local strain, but we had an hypothesis that there was a complication or something about how structural biology is done. Let me talk about that. When one goes to solve a crystal structure, you have to generate a parameter set for each of the ligands, much like if I was running a simulation, an ND simulation, I have to generate a parameter set for my ligand. The same thing happens in X-ray crystallography or structural biology. Now, how those parameter sets are generated is really quite ad hoc and variable. It can be actually a literal manual thing where people type in values and generate a file. There can be an automated way, and there can be sophisticated ways like many people do with simulations where QM calculations are done and the parameters are derived from the QM and put back in the parameter data set. So there's a whole variety of things that are done. But one of the hypotheses we had is that we wanted to test to see whether or not that parameter file format and the way the parameters are used was actually adequate to describe the ligand energy or the ligand conformation in a structure determination mode. And so here's what we did. We took this data set and we calculated the ligand energies. And I'm showing you on the left, the ligand energies for the raw PDB coordinates had an average of over 400 kcals. If we do a re-refinement where we take and generate a single parameter data set, remember these raw coordinates has a wide variety of different parameter set generations. If we take those same crystal structures and we generate a parameter set by deriving parameters from the MMFF force field. So now we have the same method to generate the parameter set for all of them. You see a dramatic drop in the energy between a mean of over 400 down to a mean of 350. We did a third method where instead of using a parameter set at all, we actually pass the MMFF gradients to the X-ray refinement package and use the MMF gradients to drive the ligand conformations. So we're throwing away the parameter file and driving these conformations with the force field directly. And what I'm showing you here in the statistics is that if we look at the difference between the MMFF gradient minimized structures and the deposited, we have a substantial improvement in performance in terms of reducing the ligand energies. And if we look at the difference between the gradients and the parameter file derived from the same force field, we also have an improvement, though it's not as large. This would be called a moderate effect size improvement. Now, because this is a session on using the CSD, we actually compared our results to using Mogul to the CSD. And one of the things that we had a suspicion is that for simple metrics like bonds, where it's a simple harmonic, if we input the right parameters, so if we derive the correct bond distance with the correct harmonic from MMFF, we would get very similar results. And that's exactly what's shown here. So you can see there's no statistical difference between the parameter file that derives the MMFF parameters and using the gradients themselves. We see essentially no difference at all when we're using a t-test. When we look at something more complex like angles, there is a dramatic difference between the parameter file trying to generate the energy and the complexity of that, and what happens in using the gradients by themselves. So in this case, our argument was, though one of the reviewers violently disagreed, that the parameter files themselves, the mechanism by which they're done, and the functionals that are used from them is actually inadequate to correctly describe ligand geometries when one is doing a refinement against diffraction data. To go back to ligand strain itself, there was a publication in 2012 where a large data set of crystal structures, high resolution crystal structures, had their local strain assessed using DFT with Gaussian. And many of the structural biologists took great offense at the numbers that were shown here because these energy values are extremely high. So Oliver Smart at Global Phasing took 19 examples from this Sitzman set. And what he did is he re-refined these structures using the same, using Gaussian and the same basis set with DFT in Buster. And he also refined these same structures using MMFF force field, like I described previously. And then determined from those re-refined structures what the local strain was for these 19 structures. And here's the result from the Sitzman paper. And here's the local strain after you re-refine these structures with the same QM basis set and same algorithm as was used by Sitzman to assess the energy. None of these structures, none of these ligands have local strain greater than 5 kcals. If we compare that to the MMFF results, you can see with one exception that one of the structures does have a difference greater than 1.5 kcals, but all other 18 structures basically have an energy that between MMFF and DFT that's within 1.5 kcals of each other. Now, why is this important? Well, in terms of solving structures, it's important in terms of compute time. So to do what Oliver did with the QM and these large basis sets took days of calculations. Whereas if he used MMFF, there was actually no difference in the time it took to solve the structure or do a refinement. So let's go through the conclusions. The bad news is There are low quality crystal structures. You have to look at the data to make sure that you're not grabbing a coprolite. And there are some bad ligand conformations. Again, we need to assess that. And Doree talked about how we can also use the CSD data to do that. Good news is that if we use the published guidelines, we can actually find the diamonds among the coprolites. And these new methods, and I've talked about new methods by which we can actually eliminate bad ligand conformations as well. I would like to close up with acknowledgements. This work was not done as a committee of one. So the iridium scoring has been done at OpenEye by Jesper, David Labard, and Bob Tolbert. The post-prediction study was done by Brian Kelly and Paul Hawkins. The virtual screening was done in conjunction with Mark McGann. The ligand strain and MMF refinement I talked about was the work of Paul Janowski, Nigel Moriarty, Brian Kelly, and Chris Bailey. And of course, I talked about Oliver Smart and Gerard Baconia in terms of the MMF and QM refinement. And thanks so much for your time and attention. And I'm ready to see who the winners are.
Great. Thank you so much, Greg, for a fantastic talk. I think there's been a burning question in the chat for about the last 10 minutes. Can you comment on the ROX AUC of less than 5 in the example you showed?
Yes, it was essentially a bad ligand conformation. So, and we know that that's the case. So we know that there's cases where the crystal ligand conformation actually generates bad ROX AUCs. Of course, the other complication with P38 is that there are DFT in and out ligands there. And if I grab a DFT out ligand and I'm trying to screen against DFT in ligands, I can't expect ROX to do well.
There you go. Do we have any other questions in the chat? I thought this was a great companion talk with you and Doree.
I'm sorry. I did have a question for Greg, which I've probably bugged him about before. Why not real space correlation coefficient in the active site as a measure of health for a protein ligand complex?
That is what is done in the iridium scoring in Spruce.
And okay, you just didn't talk about it today.
Yeah, that's what I was talking about, the 0.95 density for the active site and the ligand. So that actually is calculated now, and you can get a score. So it's not really, it's not a pure RSCC, but it is a normalized density metric.
Thank you, Greg.
Great. Well, if there are no more questions, I'm going to mute you, Brock. And I think up next is Vera Prytkova. She is a research and application scientist with the CCDC, and she is our last but not least talk of the night. So thank you so much, Vera. If you want to go ahead and share your slides.
Thank you. Thank you for the opportunity to speak today. I'm representing the Cambridge Crystallographic Data Center. I'm going to start from the beginning. So the title of my talk here is the use of the structural databases in drug discovery and how to extract useful information from the CSD and the PDB. So let me begin by telling you a little bit more about what CCDC is. It grew out of the activities of the crystallography group led by Dr. Olga Kennard in the Department of Organic and Theoretical Chemistry of the University of Cambridge. So from 1965, the group began to collect published bibliographic chemical and crystal structure data for all small molecules studied by X-ray or neutron diffraction. With rapid developments in the computing, this collection was encoded in electronic form and became known as the Cambridge Structural Database. And here is the quote by Olga Kennard that I think unites us as an organization to answer the questions, why do we exist? This is because we have a passionate belief that the collective use of data will lead to the discovery of new knowledge, which transcends the results of individual experiments. CCDC is a not-for-profit organization. It's a charity organization. We're home to 3D structural data and software for pharmaceutical discovery, materials development, and research and education. And apart from delivering the knowledge-based solutions, we also heavily promote research collaborations across academia and industry to advance the structural science. The science and research that we do mostly cover the following fields: the crystal engineering, drug design, molecular structure analysis, and knowledge mining. And if you take a look at the link at the bottom here, you can find the entire list of our publications. So I welcome you to go through them. And on the right, I'm showing some of the most recent publications that we have. So what is the CSD in the first place? As I mentioned, it's a collection of the crystal structure data. By now, it has surpassed 1 million crystal structures. All of those structures are individually available for anyone to download. Each of the structures is enriched and annotated by our experts. The amount of information that's stored in the database is really vast. It contains millions of molecular geometries, hydrogen bond interactions, and pi-pi stacks, thousands of Z'(1) structures, and multi-component systems. So you can extract information in your research that could be useful, such as mining database. You can look for interaction patterns, scaffold hops, substructure searching, or you can look for geometry and interaction analysis. That includes the intramolecular geometry, the geometry of interactions, and the likelihood of interactions. A lot of people use the CSD in their research, not just the CCDC itself. If you take a look at the citations of Web of Science from 2006 to 2016, and look at the subject category, the graph on the table on the left shows how many citations were made in these areas in those 10 years. Not only the CSD is being used by researchers, but also the CSD is enriched by the research of other people. So we take those crystal structures from various publications, and on the right, I'm showing the word cloud of common bigrams in the titles of publications that contain the CSD compliant crystal structures. So to narrow this down and go from the more broad field of crystallography to the field of the drug discovery, how can we use the power of the data? And this wouldn't just be the data of in the CSD, it could also contain the data in the PCB or any kind of database that one would like to use with the crystallographic structures. It's possible to assess the pocket druggability by analyzing intermolecular interactions between the target and the probe and construct interaction propensity maps. You can also mine databases to identify repeated patterns of interaction or potential scaffold hops. That includes also fast substructure searching of intermolecular interaction patterns. You can also go to the hit to lead stage and assess how changes affect protein ligand interaction using the CSD and the PCB and optimize compound geometry. You can check the impact of changes with isosteric replacements or scaffold hops and understand how changes affect conformation. And once you've gone through all those stages, you also would like to identify the risk of alternate forms and reveal the opportunities for intervention or provide reassurance with conformational interactions and hydrogen bond analysis. There are so many different uses, but today I'm going to focus on several more specific examples. One example would be mining structural databases to identify patterns of interaction or potential scaffold hops. Another would be using statistically significant information about molecular conformations and intramolecular interactions to evaluate the probability of observing a particular conformation of a newly designed drug in the binding site. And the third would be performing the conformational analysis of a potential drug candidate. That includes the stability analysis for solid form development. So let me dive into the first topic, mining structural databases. As a researcher, you might be asking the following questions. Which structural motifs bind similar protein binding sites? Which ligand motifs have similar protein interaction patterns? And which ligand modifications and scaffold hops are tolerated in a protein binding site? So to look more into methodology, this is methodology we have implemented in CSD CrossMiner. It's a work that's been done in collaboration with Roche. The structures are annotated with features that are based on smarts patterns. So the features could be something like the example shown at the bottom left here. So that this could be a feature such as protein, hydrogen bond donor, acceptor, heavy atom or hydrophobic feature, but this could be in fact anything that could be annotated with a smarts pattern. The feature here is represented by spheres, so each one has a tolerance radius that's reflecting the uncertainty in their position. Features positioned with respect to one another in space form a pharmacophore. So pharmacophore is supposed to represent the binding site geometry or the small molecule. And the CSD subset contains the information about the small molecule. In this case, the PZB subset will contain the information about the small molecule and the binding site. So we can mine either database with the relevant pharmacophore using the pre-built features described by the small molecule drug on the binding site or their interaction. To show you an example, this is the publication. The authors here were interested in the heterocyclic ring system produced by a three-component Gropke-Blackburn-Biennemi reaction as a possible pharmaceutical scaffold. They designed and synthesized a library of substitute products and solved the structure of one to confirm its structural properties. They then constructed a pharmacophore from this structure to identify binding patterns, to find similar structural motifs in the CSD and the PDB, and to generate new ideas for further lead optimization and scaffold hopping. To quote the authors here, the presented approach highlights the vast amount of data available in the databases and provides potential future scaffold hopping alternatives for compounds with similar binding patterns. Further studies are ongoing to introduce more drug-like properties into this scaffold. This would be one application. Another here is the molecular conformations and intermolecular interactions. And Doree has touched based on this, but I'm going to tell you a little bit more. So the questions in hand here would be, how likely is my small molecule drug conformation? Can I generate a realistic molecular conformation? What is the interaction propensity of this small molecule drug? And where is the binding site with the small molecule drug interacts? So to focus on the first thing, the CSD search for structures containing the desired fragment in a similar chemical environment, and the values of the geometries of each hit are reported. The values observed in a structure are then classified as typical or atypical of observations in the CSD by statistical comparison with the hit list. In terms of the intramolecular interaction geometries, the interrogation of intramolecular contact geometries is done by providing thousands of interactive 3D scatter plots that show the probability of occurrence and spatial characteristic of interactions between pairs of chemical functional groups. Here, the experimental data is taken from the Cambridge Structural Database, but also from the Protein Data Bank for protein ligand complexes. Interaction distributions are then displayed as scatter plots or contour surfaces, and those contour surfaces are calculated with respect to the random density based on the number of central and contact groups in the unit cell. And another application would be the binding site analysis. Here, we would like to analyze the binding site using a probe. So experimental information about non-bonded interactions occurring in small molecule crystal structures is used to show the experimentally observed distribution of functional groups or probes. So the protein binding site is broken down into structure fragments, and the scatter plots showing the distribution of a chosen probe around the structure fragments is shown. We then display the propensity maps, and those are used to aid the manual docking and structure-based drug design. To show you the application here, that would be the rationalizing the activity. Here in this publication, the authors are telling about aldose reductase. It's an NADPH-dependent enzyme that catalyzes the reduction of D-glucose to D-sorbitol using NADPH as a reductant in the polyol pathway. The inhibition of ALR2 activity may offer a promising option for the alleviation of or prevention of complications and symptoms associated with chronic hyperglycemia. In this study, the authors have synthesized several GFP model compounds and evaluated their in vitro inhibitory activity against the recombinant human ALR2. The compound on the left, epalrestat, is one of the known inhibitors of ALR2, and a docking study was conducted to look for new inhibitors. The interaction propensity maps of the binding site, in which the hydrophobic interaction area is shown in magenta, was detected using the aliphatic CH probe, and the yellow and the hydrogen bonding donor region shown in yellow was simulated with the CO probe. So overlapping the predicted docking pose of a new inhibitor into this figure clearly shows that the carboxylate group and the naphthyl ring of the ligand correlates well with the areas of predicted interaction propensity. Here we can show that the knowledge-based method is a very good addition to the docking experiments conducted by the authors. And as a third example, I'll tell you a little bit more about the solid form informatics. So the questions at hand here: is this the most stable solid form of a potential drug? Are there weaknesses which may relate to the risk of alternate forms? The selection of the commercial solid form and associated crystallization process is one of the key milestones in the development of any new drug molecule. It is critical not only from a drug substance manufacturing standpoint, but also from a drug product processing, performance, and stability perspective. The challenge for the process chemist and the pharmaceutical scientist in tackling the crystallization of highly complex new chemical entities remains a significant one. And this is because drug-like molecules often have a complicated solid form space, multiple conformational degrees of freedom, and can result in complex solid form and crystallization kinetics. Different crystal phases exhibit different surface chemistry and interaction with solvent. So I will quote the term by Bob Doherty from Pfizer, the solid form informatics and the use of structural knowledge to inform key decisions in pharmaceutical development. The solid form, the information available from a collection of crystal structure include the types of interaction that occur, the geometric characteristics, directional preferences, and frequency of occurrence of those interactions. This knowledge is fundamental to gaining the understanding of molecular lattices, such that solid forms can be assessed for stability or ultimately engineered to have desirable physical properties. To put this into the graph, you can not only assess the geometric qualities or interaction qualities, but also we look into the hydrogen bond analysis. The information contained within the structures of the CSD has been used to build training data sets for statistical modeling to allow predictions of the likelihood of hydrogen bond interactions between particular functional groups. Tools such as the hydrogen bond propensity tool allows the ranking of a structure in terms of the likelihood of its intermolecular interactions, and have found utility in the understanding of risk associated with polymorphism. So this combination of informatics-based techniques to assess intramolecular geometry, intramolecular geometry, and supramolecular interaction forms a basis of the health check. That's a term that we coin to check the solid form of a potential drug molecule. I will here represent an example, and this, as an example, we have this publication. The objective here was to demonstrate how the use of structural informatics during drug development assists with the assessment of the risk of polymorphism and the selection of commercial solid form. 3 case studies are presented that illustrate the evolution of our approach to structural informatics, and in particular, understanding the phenomenon of the polymorphism. In the first study, the authors explain how structural analysis was able to identify a metastable polymorph of Maraviroc. Maraviroc is an example of a CCR5 antagonist designed to inhibit HIV viral entry and was one of the first so-called viral entry inhibitors. The inspection of an initially designed conformation revealed that the hydrogen bond geometry was clearly unusual when compared with the other structures in the CSD. This was taken as an indication that a more thermodynamically stable form might yet exist. The compound was then crystallized in a range of diverse solvents under different crystallization conditions, and the more stable polymorph was found, thus mitigating the risk of this solid form development. In the second example, the pain candidate drug crystallized as 2 polymorphs, a metastable form A and a stable form B. The 2 forms are distinguished by both different molecular conformations and different hydrogen bonding interactions. So the structural informatics has been used to understand the polymorphism and relative stability of the 2 forms. From the conformational analysis, it was evident that the form B molecular conformation is much less likely than that of form A. Both molecular conformations represent local minima on the potential energy surface of the molecule. Stability of form B, despite its unusual molecular conformation, can be explained by its intramolecular interactions. Form B packing shell fully satisfies the expected geometry for both hydrogen bond donors and acceptors, indicating a more likely and therefore stable arrangement. Here, informatics analysis was able to quickly provide the reassurance that allows the molecular inspection to be visible for the stable form. And in the third case study, it was demonstrated how the latest informatics tools complement experimental and computational modeling approaches to provide confidence in the solid form of crizotinib, allowing acceleration of the program after promising clinical results. Crizotinib is used to treat anaplastic lymphoma kinase positive non-small cell lung cancer. It was found to be monomorphic, which means that no alternate crystalline solid form was found, even after extensive polymorph and hydrate screening. The free base of the drug was selected as the commercial solid form. So the intramolecular geometry of crizotinib was assessed using the CSD conformational analysis. All torsion angles were found to lie within the distribution returned by matching fragments in the CSD, and no torsion angle was identified as unusual. The hydrogen bond propensity showed that this structure forms a strong hydrogen bond network. The informatics analysis suggested that the solid form was stable and had a low risk of polymorphism, in agreement with the experimental findings. So the application of informatics-based assessment of new chemical entities complements experimental studies and provides a deeper understanding of the qualities of the structure. To cover what I showed today, I showed that the data stored in structural databases such as the CSD and the PDB is valuable in the drug discovery research. The databases can be searched by identifying interaction patterns or potential scaffold hops. And the information about molecular conformations and intermolecular interactions provides statistical guidance of observing a conformation of a drug in the binding site. And lastly, the collection of statistical data on crystal structure helps with the recognition of what's unusual and feeds into the risk management. With that, I'd like to thank you for your attention.
All right. Thank you very much, Vera, for a really wonderful talk. It's a really nice overview. Do we have any questions in the chat? It looks like as the night goes on, people are slowly making their way back to bed. It's a little bit late over here. Any questions? I think we might have tired everybody out. I'll leave it for a couple minutes. And then, so just so you guys know, if there's any questions that end up in the chat, I'm more than happy to save and share those. Any last thoughts, Vera?
I think that's all.
Great. This is really, really well appreciated to all three of our speakers, Greg, Doree, and Vera. This has been a really wonderful, obviously Raj is giving us claps. I can see him in the chat. Jeff as well is giving us claps. Great. Thank you so much to all of our speakers. Thank you to all of you guys for showing up. I know it's late for folks on the East Coast, but hopefully we can continue to have some of these joint BAGIM, SAGIM meetups, just because it's so nice to get everybody together where we can, while we can. And hopefully sometime in the future, we'll do it in person. But for now, we'll try to continue doing the virtual meetups. It's looking like we are likely to have speakers in May and June. And yes, absolutely. Thank you guys all for showing up, and I will hang out until people sign off. So thank you so much.
Thank you very much for organizing.
Thank you so much.
Thank you, everybody. I think we're slowly losing people, but that's good. Yeah, that was it. That was a nice set. We maxed out around 110 people. That was pretty good to see. Still apologies for the interlopers.
Rebecca,
Actually, that's the most.
this is Carmen. I just wanted to say thanks a lot and thanks for all your help too.
Of course, yeah. Thanks very much for showing up. It's I think that's the most colorful BAGIM that we've ever had, for sure.
Poor Doree. Doree will.
She handled it well. She really did.
Banned and booted. Out you go.
Did you actually flag them to somebody so they don't do it again? I don't know how you do that.
I didn't flag them so like for the future, but I have their names. So if they show back up or start acting up, I think also next time I'll probably just not let people unmute themselves. So trying to be polite, but
that's excellent. I will edit the video before we upload it.
Oh, that would be very nice.
Oh, great idea.
That would be good. All right, Brian, would you mind staying on with me for just a second? Is there anything that I need to do special to save the recording here? Because I've got it going.
I don't know. We'll find out.
I think it just uploads to the cloud. Bye, Vera.
Yeah, I think it does. And then you grab it when it's done converting.
Yeah, it seems to be set to auto upload to cloud.
Yeah, that's good.
Okay, well, we have the account, so we can grab it and hopefully it'll all work. If it doesn't, then
Great.
we'll do better next time.
Thanks, everyone. All right, I'm closing the meeting now.
Thanks,
Bye, guys.
Tony. Take care.
See you tomorrow.